In our hands, every primary AML sample that we've been able to look at has expressed some degree of CD37. CD37 has been an interesting target to work with because the ability to detect it does depend on what kind of manipulations the samples go through in the lab. So having looked at 100 samples, we are able to find CD37 on all of them in some fashion. So the work to be done is to be able to translate this into real time with patients' fresh samples to really be able to characterize that more in real time and more applicable than sort of frozen samples that we've used. But it does appear that it would be there on all samples. CD37 ADCs are a new and very exciting target we aim to explore in AML. The reason that we are so excited about this target is for many years, AML has not had very good surface targets because of the heterogeneous nature of the disease. So many different patients have different types of surface expression, and that surface expression is often also complicated by mirroring that of a normal stem cell. And so whether having a good target or a intolerable toxicity has always been something that we struggle with. With CD37, we've been able to characterize that it seems to be expressed on all AML patients. And because of that alone, this is very exciting. What's also been very meaningful is that we know CD37 is restricted only to blood cells. Yes, it is present on a number, really all other normal blood cells as well. We've been able to demonstrate that there are unique internalization characteristics to CD37 on AML blasts. So AML blasts actually internalize this antigen quickly and accumulate more and more receptors compared to all the other normal blood cells that a patient has. And so this difference creates a nice therapeutic window. I think it's too soon to say whether CD37 truly offers any prognostic data. We and other groups have looked at CD37 transcript as well as CD37 protein expression across the different types of AML cases. And in some cases, there is suggestion that it is higher expressed on adverse risk patients. So the more difficult to treat AMLs, the patients who have the poorest prognosis do seem to have more CD37 expression. Again, this needs to be validated with real-time whole blood patient samples. And just because there is higher expression, we still have work to do to understand whether that is in any way associated or causative, right, for any of the types of responses that we've seen with traditional treatment. I think that CD37 targeted ADCs offer a completely new way to try to treat this disease, one that is very exciting for patients across the mutational spectrum, patients across the ELN risk category. I think that it will be exceptionally important to collect the right data as we put this into patients to be able to understand who truly benefits.